Dear students, how are you? I hope you and all are fine by the grace of Almighty Allah in the pandemic situation of COVID-19. Here I am, M. Shahabuddin, lecturer, Department of Biology, Milestone College, Uttara. Today I will discuss from biology's second paper, that means zoology, the most important chapter, chapter 11, and I will discuss the deviation of Mendel's first law, incomplete dominance, codominance, and lethal gene. The learning output from today's class, that means what we will be learn after this class. First of all, we will be able to learn about incomplete dominance, the ratio 1:2:1. We will be able to learn about codominance, the ratio 1:2:1, and we will be able to about lethal gene, the ratio 2:1. First of all, we have to know what is incomplete dominance. Incomplete dominance is a form of intermediate inheritance in which one allele for a specific trait is not completely dominant over the other allele in condition of organism. Dear student, I repeat this, incomplete dominance is a form of intermediate inheritance in which one allele for a specific trait is not completely dominant over the other allele in condition of organism. Here are some important information from incomplete dominance. The responsible gene is known as intermediate gene. This results in a third phenotype in which the expressed physical trait is a combination of the dominant and recessive phenotypes. For that reason, the phenotype ratio will be 1:2:1 instead of 3:1. It was described first by Carl Correns in 1903. It observed in the cross between Mirabilis jalapa, commonly called as 4 o'clock plant, a cross between a pure red, the genotype capital R, and a pure white, the genotype small r, color flower plant with pink colored appeared in the F1 generation. In F2 generation, plant show red, pink and white colored condition in the ratio of red is to pink is to white equal to 1:2:1. Here the cross and the result of F1 generation, the parent phenotype red flower plant and white flower plant, the parent genotype capital R, small r, and here the gamete of this cross. And in the F1 generation, the result of F1 generation, the genotype is capital R, small r. Dear students, all the offspring are capital R and small r. That means all pink flower plants are appeared in F1 generation. Here the F2 generation of this cross, the pink flower plant, almost this is hybrid plant. This genotype of this plant is capital R, small r, and the gametes capital R, small r, capital R and small r. Here the result of F2 generation shows in Punnett square board, and we show the result here one red flower are produced, two pink flower are produced, and one white flower are produced. Dear students, this is the first deviation of Mendel's first law, and here the phenotypic ratio appeared 1:2:1 instead of 3:1. The second deviation is codominance. The ratio of codominance is also 1:2:1. First of all, we have to know what is codominance. Codominance is a condition in which both alleles of a gene pair in a heterozygote hybrid are fully expressed. I repeat this. Codominance is a condition in which both alleles of a gene pair in a heterozygote hybrid are fully expressed, with neither one being dominant or recessive to other. In codominance, recessive and dominant traits appear through in the phenotype of hybrid organisms. For that reason, the phenotype ratio will be 1:2:1 instead of 3:1. It observed in the cross between black and white Andalusian fowls, a cross between a pure black hen, genotype B, and a pure white cock, genotype W and W, yields hybrid falls with splashed white or checkered color appeared as blue in the F1 generation. In F2 generation, shows falls with black, splashed white or checkered and white color of the ratio 1:2:1. Here the result of F1 generation: parent phenotype black hen, white hen, genotype B, W and W. Here the genotype of F1 generation, all splashed white check, that means the genotype B and W. Here example of F1 generation. Dear students, you look at the picture and here shows the black and white hen can cross and checkered or splashed white are produced in F1 generation. In F2 generation, the result of F2 generation are shown in Punnet's square board or checkerboard. Here one black fowl, two splashed white fowl and one white fowl are produced in F2 generation, and that is the second deviation of Mendel's first law. The phenotypic ratio are appeared 1:2:1 instead of 3:1. Today's last topics: lethal gene, the ratio shown in 2:1. First of all, we will know what is lethal gene. A gene whose expression result in the death of the organism, usually during embryogenesis, is known as lethal gene. I repeat this: a gene whose expression result in the death of the organism, usually during embryogenesis, is known as lethal gene. French scientist Lucien Cuno in 1905 first discovered the lethal gene. The effect of lethal gene in human body, that means in human, hemophilia and thalassemia disease occur for the presence of lethal gene. This is the most important information for MCQ. In human, hemophilia and thalassemia disease occur for the presence of lethal gene. Characteristics of lethal gene. Number 1, lethal gene exists as a dominant or recessive. Yes, students, I repeat this: lethal gene exists as dominant or recessive. Dominant lethal gene is effective in both homozygous and heterozygous condition. Recessive lethal gene is effective in homozygous condition. I repeat this. Recessive lethal gene is effective in homozygous condition. The lethal gene can modify Mendelian ratio in 3:1 into 2:1. Here the some important information from lethal gene. In nature, two types of mice are available. Dear students, in nature, two types of mice are available, such as yellow and agouti. Agouti means golden brown. Gene Y for yellow is dominant over gene small for agouti color. I repeat this. Gene capital Y for yellow is dominant over gene small y for agouti color. Genotype of pure breed yellow color mice is capital Y, and genotype of pure breed agouti color mice is small y. But in nature, there are no pure breed homozygous yellow colored mouse, because in the condition of homozygous, this mice will death in embryogenesis. I repeat this, there is no pure breed homozygous yellow colored mouse, that means capital Y, and not exist in world, or they will death in the condition of lethal gene and in the situation of embryogenesis. Here, because Y gene acts as lethal gene in homozygous condition and cause embryonic death of mouse. Here, the phenotype, the yellow male hybrid and cross yellow male hybrid, the parent phenotype is capital Y small y and also capital Y small y. Here the gametes Y, capital Y, capital Y, small y, and the result of F1 generation. Here two yellow mice are produced and one agouti mice are produced, and the other mice, that was the genotype capital Y, dead in embryogenesis. For that reason, the phenotypic ratio will appear as 2:1 instead of 3:1. Dear students, this is the most important deviation from Mendel's first law, who is expressed by the cause of lethal gene, and the phenotype ratio appeared as 2:1 instead of 3:1. So, dear student, I hope you and all are understand this class, and if you faces any kinds of problems, you will contact your subject teacher. He or she will solve your problems. Homework from today's lesson, but today's class, number 1, explain incomplete dominance or ratio 1:2:1, explain codominance or ratio 1:2:1, and explain lethal gene or ratio 2:1. Dear students, keep safe, you and all, and okay in the pandemic situation of COVID-19.